is the impact of race and immune microenvironment on COVID infection severity and progression. And the team is Jason White, Isra El-Hussein, Shu, Christopher Andrews, Zainab Alwan, and Manga Tayaru Janata. I'm sorry if I'm butchering your name. So notice that some people are participating in multiple teams.
Hey, good morning, y'all. Thank you for your time. My name is Jason White. I'm a PhD student at Tuskegee University. And I was given the opportunity to lead Team 12. So I'm grateful to everybody for the opportunity. This was definitely an interesting experience, one that I would recommend to anyone. It was interesting. We kind of changed our title a bit. We ran into some problems, so we had to modify it a little bit. The new title became The Impact of Ancestry on Alveolar Macrophage Gene Profiles and COVID Infection. Sorry, I'm trying to navigate, but it's not working. There we go. All right. So we kind of already mentioned the racial disparity in COVID infection, and doing just a brief look through PubMed, you're going to come across a lot of articles that talk about, inequities in healthcare or comorbidities or social economic differences. But our lab tends to focus on the biological background of infection. We mainly investigate cancer. COVID is a new area for us, as it probably is for a lot of different people. And so we decided to tackle the question of the disparity from a biological perspective. Looking at COVID-19 infection, the early stages of the infection begin with the alveolar macrophages, and they're the resident police officers of the lungs. They're the first responders to any infection, but specifically in this case, COVID-19 infection. And so we were curious as to if alveolar macrophages were different based on ancestry, and so that triggered this particular study. Looking through the literature, what we find is that alveolar macrophages. Now, I guess I should input the caveat. COVID, as we all have probably come to learn, is new, and the research is still very limited, and so we couldn't find any data on alveolar macrophages in COVID-19 infection specifically, and so we had to use comparison infections, one of which is acute respiratory distress syndrome. We could have also looked at acute lung injury or MERS or SARS or any number of respiratory conditions, but this was sufficient to provide a starting rationale looking at ARDS. So alveolar macrophages and their transcriptional programs have been linked to outcomes of acute respiratory distress syndrome. In addition, the development of macrophages and immune response have been shown to have ancestral roots, meaning that ancestral groups share or ancestral groups have distinct immune responses and patterns, specifically where macrophages are concerned. And so in this particular study deals with GWAS, and so it was looking at mutations. However, the findings suggest that there are population differences in immune response and that are genetically controlled. And so the premise would be that African Americans could possibly possess ancestral markers within the alveolar macrophages, within their immune system at large, that determine how they respond to infection. And so how do we go about tackling this? To say it briefly, it was hard because as someone else, one of the other presenters mentioned earlier, the expression data is very limited. And so we needed gene expression data to start this particular project. Fortunately, we were able to come across a couple of data sets that were investigating COPD. within those sets, we were able to come across samples that were alveolar macrophages that were purified from Macrophage fluid. And so what we did was we took the non-smoking samples from this data set and split it by ancestry, African American versus European American, and then we compared those two groups we compared them using Lima, which in doing this we encounter some issues I'll get to in a minute. However, in doing the differential expression, we were able to show 865 differentially expressed genes out of the microarray. After doing that, we also did GSEA, gene set enrichment analysis, looking at the down-regulated pathways of interest, and I should have circled it, but you see the third from the top is innate immune response. You also have downregulation in infectious disease-related genes, cytokine signaling in immune system. And so this data would suggest that our premise was actually well-founded, and that there are some pathways in African Americans, healthy African Americans, that are associated with the response to infection. Leading edge analysis also showed the top genes that were enriched in these down-regulated pathways. We have more work to do to actually tease out how these genes, or the roles these genes play in these alveolar macrophages. However, this is a good start. We had a couple of challenges. One, as I already mentioned, was gene expression data, or the availability of gene expression data for COVID patients. It just basically, it almost doesn't exist. There's a lot of single cell papers, but most of the research done up to this point, or at least with publicly available data, is not gene expression. We also had an issue with underrepresentation of African Americans, which we spent an exorbitant amount of time trying to track down data sets that actually have race as an annotation. And of the ones that do have race annotated, what we realized was that there weren't enough African Americans in the sets to actually be usable. Another issue that we had was the fact that what we're doing, finding these data sets, secondary data analysis, leads to misinterpretation of what the data set actually is. And we actually ran into an issue where we had to redo a number of analyses to try to figure out, or we had to redo some analyses because of misinterpretation of what the data sets we were using actually were and what was in them. Moving forward, we're actually going to use this data set that we already have to do a network analysis and further tease out kind of what we're seeing. We're also going to validate these results in another data set, another COP data set, actually, that so we can see if we can validate what we found. We're going to redo the differential expression analysis. We ran into some challenges with formatting. Although the data is deposited into GEO, and GEO is wonderful, everybody doesn't deposit data in the same way. And so you end up having to kind of formulate the data differently, or format the data differently to actually get it, to process it, because we didn't process these from raw, from the raw CEL files. So we're actually going to do that moving forward. We just didn't have time in this abbreviated time period to do that. And then we're also working with collaborators to access newly generated data. There are actually a number of gene expression, or a number of studies ongoing that are going to produce gene expression data, in which we have some affiliation. So we're actually going to try to get access to that. And the collaborators specifically are disparities researchers. So we know in those sets we're going to be able to identify some ancestry related markers. And with that, I want to thank everybody. We want to thank everybody. I thank everybody for participating, the hackathon organizers, everybody that participated. I thank you guys for listening, and I appreciate everything. Thank you. Anybody got any questions?
Are you going to be able to merge the data sets or just do different analysis?
Yes, the sets we found were actually done with the same platform, so that is a thought. The initial thought was just to keep them separate to see if they can validate one another. But yes, merging them is an option.
Another question I have is, since some of the data you took were from COPD studies, I wonder if that skews the results just because there could be some different demographics of the COPD population, such as more males and more older.
Actually, the healthy samples that the samples that we took were from healthy samples. So they were just, the age range was actually pretty broad. And we didn't notice any skew. There may have been hold on, let me see real quick. There may have been a skew by gender. I'm not sure. Yeah, there were more males, but it was like, yeah, there were twice as many males as there were females.
Okay, how was the ancestry information determined? What about mixed populations or people with mixed ancestry?
That's a really good question. So I'm not sure for these studies. Typically, is the nomenclature is different. Some people say ancestry, some people say race, but most times when you look at these clinical studies, it's self-identified race is really what it is. Typically, in our lab, when we have RNA sequencing data, we will actually do admixture to determine the genetic admixture of, do that to verify the self-identified race. But unfortunately, because this was microarray data, we couldn't do that. But in future, that is something we would do by default on any RNA sequencing data set.
Final questions?
Yeah.
If not, then,
Okay. Can you hear me?
Oh, yeah, go ahead.
Yeah, I'd put it in the chat. The top gene that you found was ACO1, which is a conatase. And so this is well known to be affected and controlled by oxidative stress, which is obviously what these patients have. And that regulates movement of molecules around the Krebs cycle and would compromise the production of ATP, which seems to be a really big issue in all these patients. So was it up or down? I was trying to make notes as you went.
I can check real quick. It was actually down.
Yeah.
It was down in African American patients.
Yeah.
And what I will say is we had a number of metabolism related pathways that were affected by these differentially expressed genes. So that is an angle that we could approach it from, specifically looking at mitochondrial related gene expression. And so the next step is to kind of start teasing that apart to see exactly where these genes fit. But thank you for that.
Yeah. Thank you.